Okay, lecture 1.4, Genetic Foundations. Now we have to keep in mind that all living things on this planet have a genome. Majority of it, anything that's not viral, it's going to be DNA. Deoxyribonucleic acid. AGCT. For all intents and purposes, it's the same, AGCT, whether you're talking you, your dog, your goldfish, the tree outside, or the bacteria living in your gut. Prokaryote, archaea, bacteria, archaea, eukaryotes. Pretty much the same DNA nucleotides, the same RNA nucleotides. Within this DNA, this, set of instructions, the blueprint, as I've said before, is the instructions on how to replicate. How to replicate, how to self-repair. And it's interesting. Our friend E. coli. As I told you before, prokaryote genomes are going to be measured in millions, M. E. coli has about 4.64 million nucleotide pairs. Circular genome. This circular genome has to be able to act as the blueprint. The instructions are not only how to make and maintain the cell, but also on to make and maintain itself. Copy itself. Copy the cell, the organism itself. The DNA nucleotides, thanks to Watson and Crick, and those that they relied on, like such as Franklin, we now know the shape of the double helix, which is pretty much the same for all living organisms. Some modifications in the topographical shape, the major and the minor grooves and things like that. How much of a twist and which direction of the twist may be different organism to organism. But the replication, which is honestly, I think, why Watson and Crick really deserved their Nobel Prize, no matter how big of a-holes they were. That semi-conservative, the unwinding, the replication forks, the replication bubble that you've learned in other courses. Universally the same. Prokaryotes, archaea, us. The machinery, that's going to be slightly different. And what's really, really cool is what you see here. The central dogma. You have a gene, DNA. That gene will be copied, but into RNA, transcribed. That messenger RNA will then be read by ribosomes and translated from nucleotide language, translated to amino acid language, hence the name translation. You get the primary structure, the folding, secondary structure, tertiary structure, post-translational modification, parts of it cut off, twist, tweaks, things added, one or more polypeptides come together, and then you get the final working enzyme, hexokinase. This works whether you're talking prokaryotes, archaea, fungi, plants, animals, us.